So I would just like to thank you for organizing this conference. It's a pleasure to be here and talk to you guys about my project on this platform. Our research at UCL addresses a key unanswered issue in systems neuroscience, which we can summarize in two main questions. The first question is, what are the synaptic input patterns that are arriving on the dendrites of the cortical neurons? And the second is, how are these input patterns incorporated into the output signal of the neuron? We investigate pyramidal neurons in the visual cortex in vivo, and the cell's neuronal output is monitored by two-photon calcium imaging. To answer these questions, we need to do two things. First, to map the input arriving onto these dendrites, and second, link this input map to the tuning of the output of the neuron. For this, we need to investigate the neuronal circuit at a higher resolution. So we use electron microscopy for that. In this way, we have to correlate our in vivo two-photon volumes with the electron microscopy images. The challenge for us is the following. How do we find an already functionally characterized cell from the two-photon volume for eFIBSEM imaging when the tissue that the calcium signal originated from is now sliced up and completely black? And that's not the full extent of our problems. We had additional complexity because we are not only interested in the connections of these neurons within the in vivo circuit, the structural connectome, we would also like to know the weight of the synapses arriving onto these neurons and also the activity of these cells. So in order to identify these active synapses in vivo and estimate their weight, synaptic weight and activity, we use a specific fluorescent dye for in vivo synapse labeling. This dye incorporates into the recycling presynaptic vesicles and light up as green. The next step is to photoconvert this fluorescent dye for electron microscopy, and then to collect high resolution, close to isometric voxel size volumes. We use eFIBSEM or enhanced eFIBSEM microscopy. And for the visualization and analysis, we use machine learning based vesicle detection and segmentational techniques. The dye that we use for active synapse labeling is the FM143, which has plenty of delocalized electrons for emitting a light, and it also has lipophobic tail, which helps it incorporating into the outer leaflet of the vesicle membrane during visual stimulation. If we would look at the presynaptic terminal loaded with dye, all we would see is just a blob of greenness. Converting this dye allows us to do a precise anatomical segmentation and to precisely count every photoconverted vesicle in each presynaptic terminal, and that would be the readout of the synaptic button activity. Our in vivo experimental workflow is the following. We show drifting grating as a sensory stimulation for 10 minutes for our genetically modified animal to elicit neuronal activity. And this activity is recorded by the two-photon calcium imaging, and the data are later analyzed with sweet 2P. Then we repeat the sensory stimulation again in the presence of this FM143 dye. We also record Z-stacks, volumetric two-photon information. The first is a high-resolution initial Z-stack at the beginning of the experiment to confirm the field of view under the cranial window, and the second is at the end of the experiment, a very crude Z-stack to confirm the place and the size of the dye bolus. At the end of the experiment, the animal is immediately perfused with fixative. The next day, the animal's brain with the bolus is sliced up to 100 micron sections, and each section that has the bolus on it got photoconverted with a specific wavelength of light in the presence of oxygenated diaminobenzoate. Then the sections are processed for electron microscopy with the rotor protocol, and then we shape up our sample for the eFIBSEM imaging. Originally, we sent our samples to Janelia for enhanced FIBSEM imaging, but I'm happy to say that we have our first enhanced FIBSEM in London at the CLEM Institute, and tomorrow Chris Paddy is going to talk to you about the challenges you can face when you set up this system. So the specific L-shaped milling and imaging configuration within this microscope will allow us to mill bigger volumes much quicker than in a traditional FIBSEM microscope. This is a small sample volume from our data, and the near isotropic resolution allows us to see single vesicle in several consecutive sections. And by inspecting our data, we find two distinct vesicle classes, non-photoconverted or PC minus vesicles with a clear lumen, and photoconverted or PC plus vesicles with a dark lumen. If you look on the left-hand side, there's a button with loads of PC plus vesicles inside, indicating a high activity level. On the left-hand side, though, there are a couple of buttons where there's fewer photoconverted vesicles, indicating reduced activity for those buttons. For rigorous vesicle detection and visualization, we reconstructed axon volumes, active zones, and mitochondria, and we predicted vesicles for the vesicle detection with the help of Jan Funke and Arlo Sheridan from the Janelia Research Campus using their algorithm. And this is the output of that. The cells are different colors, the axons are different colors. The vesicles first detected, then sorted into PC plus and PC minus, red and green respectively. The active zones are yellow and the mitochondria are gray. And now that we know the aim and the workflow of our experiments, let's get back to the problem. How do we find the cell in those sliced up dark sections? We thought to use X-ray tomography because in this way we can see into the slice sections, and we can even make out the cells, and with the right resolution we can find our target cell for the final FIBSEM interrogation. The idea is not new. Carlos Bosch from the CLEM Institute already did the groundwork, and we are following his lead on this. In their workflow, they compared lab-based X-ray techniques with several synchrotron based X-ray techniques to bridge the gap between the two-photon imaging set and the block-faced SEM volume. After talking to Carlos, we decided to go with the synchrotron X-ray to increase the spatial resolution of our data, and also for a considerably reduced imaging time. So we completed a rapid application and then got to come here to the live source at this beautiful Harwell Campus and do our synchrotron X-ray computed tomography with propagation-based phase contrast. That's the whole name of the technique, at the I-13-2 beamline. In this adventure, we relied on the expertise of Christophe Raoult, Sashi, and Leonard, who helped us establishing the right parameters for the imaging. The couple of days we spent next to the beamline were highly successful, and the outcome was very pleasing. We get the right resolution for the correlation of work, and we thought we can start now finding our target cells. That's what we thought, but reality was slightly bit different. Upon putting together all the projections from the X-ray imaging, it became evident that the sections are slightly bent, and that causes a problem because we cannot now put our volumetric data simply together. So that's exactly what we needed in an already elaborated pipeline, just another step. Now we had to deform the X-ray data set for the subsequent non-overlapping volume stitching. For this, we connected the team behind WebNoSource, which is an open source tool for viewing, annotating, and manipulating 3D image volumes. They offered us a solution, and now all we have to do is to create point clouds on both surfaces on every sections, and then they would do the flattening for us. So this is what one section looks like with the point cloud, and that is the outcome of the flattening. You can see that in each view, our data is very nicely visible and straight. The next step was to provide landmarks for the subsequent non-overlapping stitching of the sections. So we placed these landmarks all over the sections, but mostly around the region of interest. That was the priority for fitting up the two neighboring volume. WebNoSource started to assemble our data together with high accuracy, and I'm showing two sections here, but at the moment we have 6 consecutive sections stitched very neatly together as a volume. The next step would be to warp the stitched X-ray volume with the two-photon volume that we collected during the in vivo experiment. And for this, again, we had to provide landmarks. This is now a bit more difficult because the landmarks cannot just stay on top of the sections. We need to put the landmarks within the volume, and that's when the free simultaneous view came really handy. This is the outcome of the warping. The two volume really nicely fits together, and you can even make out some active cells and one of the views. If we go closer, we can see 2 active cells, and if you look underneath the X-ray data set, they are just underneath the two-photon image. So our CLEM pipeline now complete, and we can go from the 2 volume to the eFIBSEM routinely from now on. And that will involve collecting the two-photon data, then photoconverting this FM dibolus, then go through the roto protocol for processing the sections for electron microscopy, then do our X-ray, synchrotron X-ray imaging, and then working together with WebNoSource, we have to do the flattening and the stitching, and then warping the two-photon data set and the X-ray data set together. Then we can go and search for our target cells, and all is left to do is to shape up the tissue for the subsequent enhanced eFIBSEM milling. In fact, we do have one volume where we have a cell that output activity is known on the left-hand corner, and its dendrite is just going across the section now. And if we have a look at the higher resolution volumes, we can see these presynaptic buttons going around this dendrite. And also what you can see is the photoconverted vesicle ratio is different in each button, which suggests that each button had a different activity during the stimulation. And this is the stepping stone where we can start answering the questions that I posed at the very beginning, how these inputs contribute to the shaping up of the neuronal output. Oh, all that left again is to say thank you for everyone who worked in this project. The core team with Anne, Claudia, Richie, and Kevin, the extended team at UCL with Mehmet, Dustin, Vincenzo, So, and Caroline. I would also like to thank our collaborators at the CRIC, Carles, Lucy, and Andreas Schaefer, Arlo and Jan at Janilia, Sean and Song, who are now at Yale, our helpers here at Diamond, Christoph Rau, Sashi Marat, and Leonard Turpin, Norman and Tobias at WebNosos, and our supervisors, Beverly and Michael. Thank you.